Hello, everybody. Today we're going to continue with our talk about the Earth's biotic enterprise. Yesterday, we looked at the drivers of evolution: natural selection, sexual selection, and chance. And today we're going to look how selection leads to evolution. And we'll also look at some chance events in future talks as well. So let's get started. What does selection actually do? Well, if you think about it, or even if you don't think about it, it changes the frequencies of alleles, and alleles are the form of a gene, from one generation to the next. For example, the allele that increases redness in male cardinals will slowly increase in frequency from one generation to the next as the redder cardinals get more of the matings, until the entire population possesses that redder allele. The change in gene frequency, which is strictly speaking we should call allele frequency, but is commonly referred to as gene frequency, from one generation to the next is called microevolution, and that's a term you need to know. Microevolution causes some alleles to be eliminated from the population and others to become fixed, which means possessed by every individual. This gives us a model of evolution. Microevolution, repeated over thousands of generations on multiple alleles, causes actual changes in the structure, the physiology, the behavior, and the ecology of an organism. Such changes include the formation of new species, but also include the major changes in structure and modes of life that characterize new genera, new families, all the other taxonomic categories. And we call that kind of evolutionary change macroevolution. Microevolution occurs on a generation by generation basis, which is 20 minutes in a bacteria to 20 years in a human, whereas macroevolution occurs over much longer periods of time, often millions of years. In fact, 2 million years is the average amount of time for a new species to evolve. So to repeat, microevolution, repeated thousands upon thousands of times, will lead to macroevolution, which are those visible changes we can see in the structure, the modes of life, the ecology, the behavior, the physiology of organisms. So we now know how evolution changes a lineage. But how does biodiversity actually increase over time? You will remember that was the second thing we said that evolution must be able to explain. So we want to understand how new species occur, a phenomenon we call speciation. So think about the point of bifurcation in an evolutionary tree. Why do lineages split? Well, they split because they have a separate evolutionary history after that point. And what that means is that two populations no longer interact with each other. Because if they were able to interact and breed with each other, then their lineages wouldn't split. So it must be that two populations stop interacting with each other. And this is referred to as reproductive isolation. They can't or they don't mate with each other, the two populations, and therefore they begin down separate evolutionary paths. Let's see how this works. Two isolated populations may be part of the same species if the populations exchange immigrants. So here we see the western deer population and the eastern deer population. Brown morphs are over here and rusty morphs are here, but deer are able to migrate in both directions carrying those alleles. Therefore, the two populations remain as a single species because there are alleles carried from one to the other. Any new allele in the western population will make it to the eastern population and vice versa. This is referred to as gene flow. As long as there is gene flow, the populations will remain as two parts of one species. If two populations have no gene flow, they will evolve separately from that point forward. And since each population will have different random mutations, and if they are in different environments, they will experience different selection pressures, they will begin to grow apart by evolution. They will have different genes, different adaptations over time. So what kinds of things cause reproductive isolation? The most obvious are geographical barriers. In other words, actual physical barriers, such as oceans, rivers, lakes, mountains, deserts, canyons, etc. any geographical feature you can think of can provide a barrier for populations. There can also be habitat barriers. For example, when a forest is surrounded by a completely different habitat, such as the grassland, which causes a population within the forest to no longer be able to emigrate, and therefore would isolate other forest populations of what was once the same species. When two species arise because of physical separation due to geographical barriers, we call it allopatric speciation, which means separate homelands. Here's a picture of it. Here is a population of insects. They get divided by a river, and over time, the two populations diverge. Here's an actual example of allopatric speciation. The Isthmus of Panama formed about 3.5 million years ago, and when it did, it separated some ancestral porkfish, great name, Anisotremus is the genus, and as a result, these two populations have been diverging ever since. The Caribbean side, which is now the porkfish, Anisotremus virginicus, and the Panamic porkfish, Anisotremus tenuatus. That's an example of allopatric speciation. There are other forms of speciation. Speciation may occur when the ranges are not separated, and this is called sympatric speciation. Here, female cichlids, which is a kind of fish, have a genetic preference for a certain color male. Some females like orange males, and others like blue males. Earlier, we talked about sexual selection and arbitrary choices, and indeed, these two types of females seem to be making arbitrary choices. The females don't make many mistakes, and as a result now, the orange male population and the blue male population are separated. There is not gene flow between them, and they are beginning to diverge. They are on their way to becoming different species. We know this because of an amazing experiment that was done where the male and female fish were put in tanks with monochromatic light so the females couldn't tell the color of the male. And then she mated with either color male. It was only when she was in normal light that she would choose the male that she genetically preferred. And since they live in the same place, this now is sympatric speciation. We have another example of sympatric speciation with the apple maggot fly. Apple maggot flies feed on apples, go figure. And they were introduced to North America about 200 years ago when apple trees came over from Europe. But some of them started to use the native hawthorn fruits. Let's review some fly biology so you can remember the fly's life cycle, in case that's not in the forefront of your thoughts right now. Flies begin as eggs. The eggs hatch into little maggots, which live in the fruit and develop. And then they become a pupa. I should have put a pupa in there. And in that pupa, they metamorphose into a flying adult that mates and then lays eggs. And that's the life cycle. Females typically lay eggs on the type of fruit they grew up in. So if you grew up in an apple, you're going to lay eggs on an apple. And if you grew up on a hawthorn fruit, you're going to lay eggs on a hawthorn fruit. And males look for mates on the type of fruit they grew up in. So if you grew up in a hawthorn apple, you're going to cruise for mates on hawthorn apples. And if you grew up in an apple, you're going to cruise for mates on an apple. We call this assortative mating because they're not mating randomly. They're choosing the fly with which they mate. As a result, flies raised on hawthorns usually end up mating with other hawthorn flies, and those raised on apples tend to mate with flies that were raised on apples. Now, here's the other thing that's going on. Fruits on hawthorn and apple mature at different rates. So the flies have started to evolve different timing in their life cycle to better track the species on which they feed. And now gene flow between the two populations has been reduced, and sympatric speciation is occurring in less than 200 years. And so that's another example. And it is thought that in general, insects do a lot of sympatric speciation by this process of specializing on different hosts. So what happens when two populations begin to diverge? They can either continue separately and become two new species, or gene flow can cause them to remain as one species. Those are the two choices. And so it must be in evolution that a lot of lineages start to split and then come back together due to gene flow. But at the same time, a lot of lineages start to split and never come back together. Now, in each species, other traits may evolve that coincidentally decrease the likelihood or the ability of the two populations to make a successful offspring. Such traits are called isolating mechanisms. The point about these mechanisms is that if they occur, they increase the chance of speciation occurring. These mechanisms are commonly divided into prezygotic mechanisms and postzygotic mechanisms. Let's go back to intro bio and figure out what that means. The zygote is the fertilized egg. So a prezygotic mechanism is a mechanism that prevents a physical mating. And a postzygotic mechanism is a mechanism that works against the offspring that is produced. So let's look at the different types. So here we can see a temporal isolating mechanism would be the two species might mate at different times of the year. Their timing is different, and that's what we saw happened in the hawthorn flies and the apple flies. Their timing was becoming different, and that was becoming an isolating mechanism. We may also see a postzygotic mechanism such as hybrid infertility. A hybrid is produced, but that hybrid is unable to produce gametes, and therefore it is unable to mate and produce another generation. You can take a look at each of these isolating mechanisms and see if you understand it. If not, please come to class with questions about them. Anyway, let's move on. If the hybrid itself is perfectly fit, it has no survival issues and can mate with other members of either population, then the two populations remain as one species. But if the hybrid is not fit, if the hybrid has any sort of reduced ability to mate, reduced ability to produce viable offspring, then any isolating mechanism that occurs by chance is going to be heavily favored, because individuals that make mistakes will not have their genes passed down in perpetuity, and individuals that don't make mistakes via one of the isolating mechanisms will be selected for. Once the hybrid is less fit, then over time, postzygotic mechanisms should be replaced by prezygotic mechanisms because of the big cost of making a mistake and breeding with a member of the wrong population. So that's how isolating mechanisms work in speciation. So now we want to go on and just say, what is a species? Perhaps the most commonly known definition is that a species is a population of organisms that can interbreed or potentially interbreed with one another, but cannot breed with other members of closely related populations. So if you can breed with all the members in your population, you're a species, and if you can't breed with the members of that population over there, they're a separate species from you. This definition is based on the behavior of populations and not on similarity of appearance. So it allows for variation to occur in the population, and it allows for change to occur over time. This idea is called the biological species concept, which we'll abbreviate as BSC. Some of the problems with the BSC is many populations don't interbreed because they are separated by geography, but they could reproduce if brought together. In many cases, these populations have evolved distinguishing characters that demonstrate they are beginning to have separate evolutionary histories, and that the tree of life has begun to branch. And so many systematists now argue that such populations are in fact separate species, and they call this the phylogenetic species concept. The phylogenetic species concept defines a species as an isolated population that can be distinguished from other isolated populations, even if they could potentially interbreed. That means that if an individual can be diagnosed as belonging to a certain population, if we can look at it and say, oh, that individual there came from the population on the east side of the Andes, and that individual there came from a population on the west side of the Andes, then they are two different species, unless there is compelling data that gene flow is occurring. The problem here is that there may be very low levels of gene flow connecting the two populations, and they don't have an effect in a human time frame, but they may have an effect in the long run. Or maybe in 200 years, there'll be gene flow that will swamp the changes that have already occurred, and they will revert to being a single species. That's an argument that's going on. It's a little esoteric. I'm not going to go any farther in it. If you're interested in it, I can point you in the direction of looking at that. What's the best species concept to use? Well, it really depends on what group you're working with. If you work on a group where gene flow can be ascertained, then the biological species concept is really good. If you work on a group where it can't, you might use the PSC. There are other species concepts I'm not going to go into. Most insect taxonomists, for example, use a morphological species concept. The big problem here is species concepts matter for conservation. The phylogenetic species concept creates a lot more species. Under the biological species concept, there are about 9,000 species of birds, while under the phylogenetic species concept, there are at least 10,000 and maybe as many as 15,000 species. In one study, the phylogenetic species concept resulted in an average of 49% more species. That's almost 50% more species being recognized across various groups, mainly vertebrates. This will mean that there'll be many more biological populations that warrant conservation because these are now smaller populations, they have lower numbers, and therefore they're more likely to be endangered. Our interest in species began with that bifurcation in the evolutionary tree because this is what increases biodiversity, and we wanted to understand biodiversity. So now we understand that if a population has a separate evolutionary trajectory or path from a population of similar organisms, then it is a species. Speciation occurs when they become reproductively isolated for long enough that they have measurable differences. And being able to diagnose and count species is important because we now know that more species means more biodiversity, and that more biodiversity is in turn associated with more productivity and more stability over time. So that's it for today. Tomorrow we're going to look at where species are located in the world, and then we're going to go on and look at extinction. Until next time, study hard.